Now I would like to introduce Alba to the floor. She'll be presenting using the IEDB, an analysis resource for identifying candidates in SARS-CoV-2. Alba has been doing some amazing research with Alex and many other people in the lab, so we're very excited to have her present. And Alba, if you'd like to take a little bit longer than your 15 minutes, feel free, we do have the time.
Okay. Well, then while I prepare my presentation, I will start to say that I am an exciting user of IDB. It's been 10 years I've been using this website. And actually, the knowledge of IDB, together with the knowledge of the cool science that Alex Sette was doing, was the main reason why I applied for a postdoctoral position in Alex's lab 5 years ago. And I'm still working with him very happily. One thing I wanted to mention is behind this, they are all fantastic scientists, but also very, very nice people. So it's very nice collaborating with them.
So, congratulations, Alba. That's a great milestone.
Thank you. So can you all see my screen?
Yes, all good.
Okay, so in order to guide you through the rationale of our studies, I will have to ask you to go back in time, late January of this year. So there was a novel coronavirus, there was an epidemic going mostly in China, and it was very concerning, expanding rapidly. There were no information available because this was a novel virus overall, and the sequence of this virus was just being isolated since a few days. So one of the most pressing questions that we were having was whether we could detect an immune response against this virus, as that was a fundamental point for a vaccine strategy. And because there were not information, the first question that we ask is, can we see if this virus at the protein level is similar to other family members of the coronaviruses? So to address this question, we actually performed collaboration with the VPR databases. And thanks to their very nice analysis, we realized that indeed this novel coronavirus was the most similar to SARS-CoV, which is one of the reasons, together with other studies, why it has been renamed SARS-CoV-2. That also opened a very interesting point, which is, can we use information performed on this previous virus that have been studied in the past years to actually infer potential immunodominant region relevant for this new novel coronavirus? And I guess between Randy, Bjoern, and so forth, they did a fantastic job. So I'm not going to go too much into that, but we perform a query, try to dissect the different components of the immune system we were interested in, CD8 T cells, CD4, and B cells. And we were majorly interested in looking at the response in human, particularly for the T cells. That was a requirement. So in the results that we retrieved from this analysis, there were many information that we were interested in. The first one, of course, was the list of the epitopes that we were able to extract. And that was also important because I would like to remind you that IDB is live, like in all sense. It continuously updates with very interesting information. So it's also important to store at the moment of your query and perform this query multiple times because you can always find new things that can drive your science. Together with the list of the epitopes, we were also interested in understanding, considering this was a virus with a lot of proteins, whether we could pinpoint to activity to specific antigen. So that was already an information that we wanted to understand. And also we wanted to understand within the specific antigen if we could detect specific immunodominant region. From an experimental point of view, it was also interesting for us to understand how these epitopes have been identified from an assay point of view and also read more in the relation of the references. Instead of doing multiple queries, this is all available in IDB, so that's very convenient. The results of our analysis show indeed that the majority of the epitopes in IDB were on SARS-CoV. That was fantastic. It was the most similar viruses. And also several epitopes have been previously identified for B cells and T cells. When we look more in depth to the antigen that were specific of these epitopes retrieved, indeed, 3 major antigen were shown, the spike, nucleocapsid, and membrane protein. So overall, based on our first analysis, we realized that there were B and T cell epitopes available in IDB for other viruses. Hence, we could try to use this information to infer potential immunodominant region in the SARS-CoV-2. And to do that, we extract the epitopes from the IDB, and in this case, we use still the ImmunomeBrowser, but the standalone tool that allowed us to add the epitopes of the different coronaviruses and map it to the SARS-CoV-2 FASTA sequence that we had at the time. And the results of this analysis allowed us to identify 10 most immunodominant regions encompassing the spike membrane nucleocapsid and with high sequence identity, therefore suggesting that those could be promising candidates to be evaluated. But together with the information available in IDB, we also ask ourselves whether we could utilize a prediction strategy in alternative to actually define potential additional immunodominant region for the B-cell response and the T-cell response. So in the context of the B-cell response, of course, we were interested in both in continuous and discontinuous epitopes. And in order to achieve that, we actually utilized the two different prediction systems that Bjoern went over before. The idea at the time was to analyze the spike membrane nucleocapsid, because based on previous data in the SARS-CoV viruses, those seem to be the most relevant in terms of antibody response. And considering that in terms of crystal structure, only the spike protein was available while the other three, they weren't, that was also a good way to actually investigate the three candidates. And I'm not gonna go too much into how you query. Bjoern made a fantastic job already. As you pinpoint, you can adjust the threshold of your analysis, and we decide for a threshold a little bit more conservative. We want to be more specific in this case. But of course, the type of threshold you select, it really depends on the scientific question you're interested in. For the structure-based prediction, instead, initially we utilized a homology modeling model because there were no crystal structure available for the spike of SARS-CoV-2. And in between our analysis, we were actually able to repeat the analysis with the crystal structure. The overall output, whether you use a PDB model or a crystal structure, doesn't change. And indeed, we were also able to nicely pinpoint on the tree structural level where the epitopes were located. I'm not gonna look into the different results for the proteins. I'm focusing only on the spike, but overall, we were able to identify several continuous and discontinuous epitopes in the spike protein. And throughout the months, several studies have been published validating part of this prediction. So that was very nice. Our lab, and particularly my work, have been focusing way more in dissecting the T-cell response. So I was, of course, eager to dig more into that aspect. And I'm sorry, can you all hear me?
Yes, we can still hear you and see you, Alba.
I beg your pardon, there must have been something with the presentation. Okay, now I got the replenished. Apologies. I'm gonna share my screen immediately. Sorry about that.
Oh, no worries.
Okay, can you see my screen moving?
Yes, you can see it now.
Excellent. So going back to the T-cell prediction analysis, the question that we were having is to actually try to identify immunodominant region, but also give a look into the fact that the pandemic was spreading rapidly worldwide. So we want to make sure that we take into account the HLA variability that is present across the different populations. Because of that, the type of prediction algorithm that we utilize were actually heavily focused on this aspect. And in the context, for instance, of the CD8 T cells, we select an algorithm that is designed to actually allow promiscuity. What I mean with that is if you have the entire proteome and you actually perform this algorithm on different HLA alleles, you try to pinpoint which regions are the most recognized across different HLA class II molecules. That suggests that actually those are going to be immunodominant regions across the different population, and the likelihood that these epitopes are going to be presented by the immune response is actually higher. Following a similar strategy, we also perform a CD8 T-cell prediction. In this case, we utilize the NetMXC PAN 4.0, as Bjoern suggested, is actually now the gold standard in the IDB. And always with an eye in terms of the population, we decide to select the 12 most frequent HLA class 1 alleles. So from an experimental point of view, that allowed us considerably to reduce the complexity of the virus. We went from 10,000 amino acids to actually roughly 250 epitopes for the CD4 and roughly 600 for the CD8. But that was not sufficient for us from an experimental point of view because there were still a lot of peptides to analyze. But this is the reason why, and I beg your pardon, just delayed, but this is the reason why we actually perform the megapool approach. The megapool approach is an approach that has been performed in our laboratory to actually take into account the fact that you want to test multiple peptides, but often you are limited in the amount of cells you have, or you have the problem of the DMSO toxicity. This is why, from an experimental point of view, we perform sequential lyophilization. Therefore, the megapool has the smart filtering of taking into account all the antigen of the virus, taking into account all the most frequent HLA variants worldwide, but also it allows, from a technical point of view, to reduce the DMSO toxicity, but also it achieves a synergistic effect. What do we mean with that? We mean that if you want to look at a single response on a epitope, sometimes you need to perform in vitro stimulation to actually be able to detect this response. When you combine together all these epitopes, you're actually able to dissect the T-cell activity ex vivo. So that's very important, particularly if you want to do immunophenotyping. Does it work? That was like the million-dollar question. Also, we wanted to understand indeed if we could detect T-cell response in COVID-19. And indeed, because the vaccine strategy were heavily focusing on the spike protein, we decided to analyze the spikes separately from the rest of the genome. So as you can see, we do have a significantly higher response between unexposed and COVID-19 donor for the spike. But we do also see a very stronger activity for the remainder part of the genome. Therefore, that suggests to us that their activity against SARS-CoV-2 in the context of T cells was not only focused on spike, but there was way more behind it. We decided to actually dig more into this point, and we actually perform an alternative strategy where we analyze the entire proteome of the virus. We were also curious to understand how our large-scale analysis was comparing with the T-cell prediction. And indeed, there was a very nice positive correlation. And in particular, the algorithm that we select was supposed to capture 50% of the response. And indeed, in this context, we captured 50% or even more of the response. Similarly, we see a very, very, very nice response in the context of CD8. 70% of our donor actually responded to our megapool. And just to point out, the reason why you see CD8A and B is just because the megapool overall was composed of 600 peptides, so we have to split it into different because of technical reasons. So with that, I would like to thank you for your attention. Apologies for a couple of issues in the presentation. There are so many people to thank about this project. Definitely the collaboration with IEDB, the VPR, and many people at LGI have been determinant. And I would like to thank them all for the support in actually progressing in this analysis. And if you're interested in dig more into the story that I've presented today, they're actually summarized into different papers. One focusing more on the prediction analysis and the other one focusing more on the experimental validation. With that one, I would like to thank you for your attention. I'm happy to take any question, if any. Thank you.
Excellent. Thank you so much, Alba. I've heard parts of your presentation a few times now, and it's always very, very interesting. So no, thank you so much for your time. And as I mentioned, we are running slightly ahead, but again, it just means we can answer more questions. So please feel free to all of our attendees. We're getting towards the pointy end of the workshop, so submit as many questions as you can. because now I would like to invite Alex and Bjoern back to the stage, and they will start taking some of these questions on. So thank you, Alex and Bjoern.
Yeah, so I think as I did the first presentation, I think the first questions are primarily for me, and then the later ones, Alex and Alba, you should look into those, and I'll just start with the very first one. But obviously, I mean, feel free to answer anyone in between. So the first question was, how does the benchmark work? Is it possible to download the data set for the benchmark? And I'm going to actually type some links in there. So there's the benchmark is published in PLOS Computational Biology of this year. It's relying on a data set from Nathan Crofts and David Sharkey's lab on vaccinia virus, which has been a fantastic data set because there we actually had data available on a comprehensive study of testing all reasonable candidates for T-cell reactivity. There was MHC ligand dilution data available where the comprehensively was looked for what actually peptides are naturally processed and presented by MHC, and a lot of MHC binding data as well. So that's been our model system that this last benchmark that I showed was working on. We've done other benchmarks as well. They also linked to on the site, but I'll post it in here. And yes, the data set is available, and actually also the code that produces the metrics is all on the public GitHub repository. So I'll post that on the answered question right after this as well. Alex, you want to take the next one? Or do you want me to continue?
Yes. So I'll take one. There is one question that was: do these tools account for Treg versus T cytotoxic epitopes? And I think that's a very good question. That goes back to the fact that in reality, what the IEDB curates and what the IEDB does is to predict epitopes, and whether an epitope may be associated with a regulatory function or a cytotoxic function or a secretion of particular cytokine is usually not something that is ingrained in the sequence of the epitope. It might depend completely on the context. So if you have an epitope and you inject that together with IL-10 in a self-tolerance system, that may be inducing a regulatory function. While if you induce that with a toll receptor 7 agonist, that might induce a completely different response. So it's important to keep in mind that the biological function of epitopes is dependent on the context. Bjoern, do you want to pick another one?
Yeah, I'll continue from the top. Would it be possible to have an option to choose more than one prediction methods before running the binding prediction instead of running several times, one method at a time? That's a good point. So currently on the main website, you cannot do that. On the API, it would require a loop. That's fine. Same with the downloadable. But we are actually working on a revision to the analysis tools. And Jay is going to present that tomorrow. And when we do that, there will actually be the ability to run multiple prediction methods at a time. Because in the end, it looks very similar, right? You get like another column essentially of output. And that would then avoid having to run multiple times and merge it back together. So it's something we're working on, but I would not anticipate to have that done before mid-year.
So I will answer a question regarding the, towards the end protein in SARS-CoV-2. And so the question is that since N protein is mostly intravirus, I've been discussing, not me, the question asked, but I've been discussing with some collaborators when it's meaningful to predict for antibody epitopes, since then it would not be not be shown on the virus surface. Neutralizing antibody would not work then, right? Well, that is also a very interesting question. As far as I know, antibodies against N are commonly detected, but they're not neutralizing at least as a first approximation. The neutralizing antibodies are classically directed against spike. So beyond that, if I can speculate, what would happen if you predict antibody sites for the N protein, you would predict antibody sites for the N protein, which is made of an N and a C terminus with a connected link. And so it's an interesting question to wonder. You will probably predict epitopes that are predicted to be expressed on the N or C terminal surface, which is great. Now, are those N and C terminal domain surface accessible in a virus from the outside? Maybe not. So the question is, it's on the surface of what? It's on the surface of a protein? Yes. It's on the surface of a virus? Maybe not. So that's kind of a, I know that I did not answer your question completely, but it illustrates how the virology and the biology of a virus is a higher level of complexity beyond predicting what is likely to be recognized on the surface or something for antibody responses.
Okay, I'll take the next one. When I'm seeking B-cell structural epitopes, is it possible to search for protein glycan epitope as in the anti-HIV PGT antibody? Yes, it is. So you can specifically search for, in our case, this would be considered like an amino acid modified residue. And you can specifically search for those. And then obviously you would couple that with looking for antibody B-cell epitope. And then if you're looking for a 3D structure specifically, you can also check for that 3D structure. So that's kind of a query question, not a tools question, but I thought I would answer it.
So there is another question. Can you explain how cluster analysis is different from EpiVax cluster? My understanding is that the IDB cluster epitopes from different sequences based on homology, where cluster identify cluster of epitopes within a single protein. So first of all, as a disclaimer, I don't know how EpiVax cluster works because I never used it. But my impression is that you are correct and that that tool identifies clusters of predicted epitopes or predicted binders in the sense of cluster of predicted things for different HLAs on a protein sequence. Our cluster tool has no relationship whatsoever with that, in the sense that our cluster tools clusters sequences without having any preconceived notion of whether their clusters map to a single protein or they could be derived from different sources. As I showed you, you can use to cluster wrapper, supper, daytime, and anytime, and whatever sequences that it's a mathematical operation establishing how different sequences, strings of letters, can be clustered together. And that can be applied if things from a different, the same protein, or coming from related viruses, or whatever. So it's a different notion.
If I just may add one thing to this, because Alex, not everybody necessarily knows EpiVax here. The fact that Alex just said he never used that tool isn't because he's dismissive or particularly doesn't like them. So EpiVax is a company that is selling a suite of tools that is very similar to what the IEDB provides. And people often ask us about comparisons of the tools. Our tools are all publicly available. They can be downloaded and compared. We don't have that access to the EpiVax tools, so we aren't able to provide these comparisons that people are asking for. That's not because we don't want to. It's just a difference. And it's obviously understandable. If you're trying to sell something, you need to keep it somewhat secret. So let me do the next one. Can we use prediction tools for human cancer neoantigens? Absolutely. So the tools, as we have shown you, have been developed based on data on the IEDB, and they haven't been specifically developed for human cancer neoantigen. But at least as it comes to things like peptide MHC binding, an MHC molecule doesn't know that the peptide sequence is related to cancer or a virus or whatever. It's molecular interaction. There's no reason to believe that these would be fundamentally different. And that we've published a few things on that, and I think that's pretty clear. Now, there's going to be considerations that might make some epitopes more or less relevant, and there's going to be cancer-specific aspects to it, because it's obviously a very different type of immunological context of that recognition. But there's nothing wrong at all, definitely when looking at some things as binding, to use the existing IEDB tools, essentially molecular interaction.
So one question says, can you use the IEDB to obtain HLA distribution data, percentage of people in a population that have particular HLA alleles, or do you need to use particular epitopes as shown in the presentation to see a population distribution? So funny you should ask. Actually, Alba and I were together with John Sidney putting together some tables just the other day. So the tool is not meant to do that, but there is a way to cheat and extract the HLA frequencies just by looking at the top. And if you want, you can email me, and I can, or Alba or John can get back to you and give you some detail. But basically, yes, and also with a disclaimer that that data is not our data, but we extract the data from a resource, HLA frequency net, whatever, which is a very nice resource, but it's not ours. We don't maintain that.
So the next question we have here is, it seems that currently the API tools are run on Linux OS only for the local version. Are there any plans to be able to run the API tools on R and Windows at some point? So, so I want to distinguish here. The API tools, the API are the things that are called through the web server. So at the moment you're going through the web server calls, you can run that anywhere. That's going to work in R and Windows. They can run that. Actually, there was another question. That could be done by VBA on Excel, and anything that allows you to run any kind of URL connection to the internet will work with the API. The downloadable executables are Windows, Linux only, and there's no plans to port that. And the reason is a number of dependencies there to a large set of libraries that just don't exist for Windows. So we're nowhere, and also actually for Apple. So we have issues with several of these things, and actually already maintaining the downloadable tools as they are is not trivial. So unfortunately, there's no plan to provide another API. If you have specific problems doing that, there are a number of workarounds how to do calls in a virtual environment to a Linux box within a Windows environment that we can help with. But I mean, normally I would just recommend using the API.
So there is a question that says, do you think the inability to detect cross-reactivity of memory T cell challenges the frequency of immunity among common viruses? Is this the case SARS-CoV-2? So I'm not 100% sure I completely get the question. So I will answer what I think about the topic. In general, there is cross-reactivity is dependent on sequence similarity between two different sequences. And that has been shown to be relevant in a number of different systems. So there have been a number of investigations. For example, Bjoern mainly produced some very nice data showing that in the case of allergens, if you have an epitope that is, say, from Timothy grass, and is also conserved in Bermuda grass and some other grass, then people tend to focus on those epitopes just because you get re-exposed by that epitope over and over time. The same has been found to be true in the case of hemorrhagic fever, where dengue, different substrate of dengue have sequences that are similar, and so the repertoire evolves towards recognizing those. And recently in SARS-CoV-2, it has been shown that indeed there is some degree of pre-existing immunity in the general population against SARS-CoV-2 sequences, despite the fact that these people have never been exposed to SARS-CoV-2. And this has been shown to be related to sequence homology to the common cold coronaviruses, as Alba was highlighted. Whether this matters or not is still an issue of debate. It could be that people that have this cross-reactive immunity have an advantage because they mount a faster response and thereby would have decreased severity of disease. We don't think that T cells can prevent infection. You need antibodies to prevent an infection. But once an infection occurs, if you have a pre-existing T cell immunity, there could be an impact on severity. And in fact, myself and Bjoern in other studies in relation to the swine origin flu pandemic had shown that indeed there was this cross-reactive T cell response, and this mapped back to actually people that were exposed to a casing on an H1 that circulated several decades before, had still got infected, but had lower disease severity. So it's a complex thing, but cross-reactivity between related viruses and antigen does matter, but in a not completely predictable fashion.
And I think, Nina, that answer actually took care of the first question as well. I think that was just an incomplete version of it. So I was going to go for, are the tools API compatible with R? So I would answer that with yes and no. The API runs again through just a web call, and that it returns anything that allows you to run the URI with like that do the query parameters that you want, and then as a result you get a JSON object back, and every language that I'm aware of and every environment I'm aware of able to parse that. But you will have to do some coding yourself around that, parse the payload, and with some JSON library. So if, I think your question is also a reminder to us of something we had been asked for several times before. We might want to write kind of like a wrapper around the API for the commonly used languages, which would be primarily Python and R at this point, to allow people to not even have to look at all how the URI call goes, and then just work with data.
So there is a question in continuation to Rob. It is possible to link IDB with allele frequency database for ease of selection of intended population in the future. So the IDB is linked with allele frequency database resource, as I was mentioning. We're not maintaining any HLA frequency database, and so the link is kind of behind the scenes, if you wish. I think maybe what you are suggesting is to do to modify the HLA frequency population coverage tool to allow to display more easily the HLA frequency in different populations. If you just want to know HLA frequency and you don't want to do a population coverage, that's a good suggestion. We'll take that into consideration. Thank you.
Lucky, I got a second question. Okay, so Leopoldo, your question says something about Frank scores, and I don't know what they are. If you could maybe specify a little bit as a follow-up question, but I'll go with the first one. Related with cancer neoantigen questions, do prediction tools consider the self-antigen leaning of immune cells? How can we determine the immune-activating neoantigens which can escape from self-antigen learning? So that is actually something we have looked into quite in detail. You would be expecting that mutations in cancer that are very close to the cell are less recognized than the ones that are like more disruptive mutations. And so essentially by looking for how different is an epitope from cells you're expecting to have a higher immunogenicity, and we do find that. We are actually in the process of doing some implementations of that. And you could envision the same kind of rule should be true for also viral antigens or so. Yeah, so currently those are not online. And as a heads up, this has not been as dramatic as we were hoping or expecting. There seems to be, actually, the immune system seems to be quite capable of recognizing peptides with single mutation to self very effectively. And you think about it, that actually, given that the vast majority of cancer mutations are just single residue mutations, it's not that surprising that needs to be possible, because otherwise we would all be dead. Okay. So I hope that was not very satisfactory, but at least complete answer. So now we have the Frank question. Alex, I don't think you know what that means. Okay, here it is.
Yes, I have no idea what Frank scores are. I
it's
acknowledge my
rank. Okay, sorry, Frank, and I think Frank is just fine. No, I think that's the answer. So Frank scores, as is the metric that was used to essentially like rank predictions and look like where is your prediction falling, like in a rank of random ones, which is a way to, it's an alternative measure of performance, which was purely implemented as part of the associated papers. We don't have any F-rank implementation on the IEDB side itself. And I think that covers it.
Yeah.
Yes, I think that's it as well. Oh. Thanks, Leopoldo. Excellent. So they are all the questions for now. Thank you, Alex, Bjoern, Alba. Thank you very much for that. We're only running about 5 minutes ahead. So I will move on into the closing remarks, do a little bit of a wrap up, and discuss the feedback survey, which is now again being populated into the chat. So I did share the feedback link at our midway point, but I've just shared the link again. If you'd like to open it up and start filling it out while I do the closing remarks, that would be fantastic.